There are a million different ways you could calculate biological age. You might look younger or older than you actually are. And that could be the same for your dog. When we start thinking about biological age, we want to make sure that was something that would be useful in general to pet health.
Eventually, a cheek swab will help us understand if this dog is headed towards not just any disease, but specific diseases.
The more we know about that, the more we can course correct before we're ever developing a disease. So that way we're working to avoid disease development in the first place. If you can solve for not developing the disease in the first place, that's where you really move the needle on longevity, because you're preserving that health span.
Hi, and welcome to the Dog Longevity Project. I am your host, Dr. David Haworth. I'm a veterinarian with a PhD in physiology and a real passion for the bond that we have with our pets. I've spent my career exploring all the different ways we connect with our pets, and with this podcast, I'm talking to some of the brightest minds in the field about ways that pet owners can help their pets live the longest, fullest versions of their lives possible. This podcast is brought to you by Panio, a platform designed to bring together all the information that you have about your pet, their diet, their lifestyle, genetics, health data, and then help you make steps to keep your pet healthier and longer. Today, we're going to be talking to Andria Beal, one of my very good friends who is also a foremost expert in the epigenetics associated with our pets, and specifically the epigenomics associated with aging. She is the founder and CEO of EpiPause, a leading company in the pet space about genetic and chronological aging. Andria, welcome.
Thanks, David. Hi, I'm excited to be here.
Me too. So let's start, if we can, just at the very basics. When we talk about epigenetics, what are we really talking about?
So epigenetics is the study of molecules and mechanisms that are interacting with the DNA itself, often turning genes on and off. So epigenetics is what helps organisms adapt to their environment. Like you walk into a room that's a different temperature, your epigenetics are going to change to adapt your body to encounter that new temperature. Same with anything that you're interacting with in your environment. Epigenetics is playing a role in that adaptation to it.
Cool. So like DNA is the blueprint or the book of life, but epigenomics is sort of determining which pages and sentences that we're reading and when.
Yes, exactly. So another way to think about it is we're complex organisms. We have different cell types. Every cell has your whole entire blueprint, your DNA code. But your liver cell and your skin cell look totally different. And that's because of epigenetics. Yeah, thank goodness. That's why I always say is like, you don't want your eye doing liver functions, or something like that. I don't know if it'd be cool.
At least not quite as useful as the liver is. I like my liver doing exactly what it's supposed to be doing, and I like my eyes doing exactly what they're supposed to be doing. So what kind of molecules and mechanisms do this? Are there thousands or are there just a few? Like how does the genes get turned on and turned off?
Yeah, so there's a couple of major ones. So DNA methylation, that's the one we know the most about, and the one I'm most familiar with myself and I work with as a scientist. But then there's histones, there's small RNAs, microRNAs, there's histone variants. There's all these things that are interacting. So there's a handful, I would say, of different molecules.
Cool. And so you said the DNA methylation is the ones that we know most about. Could you say more about what that means? What's a methyl group?
Yeah. So we have it's this little chemical molecule that's attaching to the outside of the DNA. And in particular with DNA methylation, if you're familiar with the DNA sequence, we have A's, C's, G's, and T's. In that sequence, and C stands for cytosine. That's where these DNA methylation groups attach to. And then they're functioning and turning that gene on or off or helping support the expression of a gene.
Oh, that's fascinating. So is this happening in every cell all the time, regardless?
Oh, yeah. So there's, in mammals in particular, a majority of cytosines will be methylated. And then it's certain areas of the gene. So getting a little bit more into the science, we have promoter regions, which are often functioning and turning that gene on and off. That's where we typically find the methyl groups that are directly related to if a gene is on or off. But we're also seeing that it could mean the same. It could have something to do with the expression in other areas of the genome, but it's a little harder to study.
Yeah, I mean, nobody's ever going to claim that genomics is simple, but it's funny because it does kind of boil down to certain repeated things that happen. Like there are only 4 letters in the genome, and
Yep.
cysteines are always the ones that find the methyl groups find, lock on, and then depending on what that cell is doing and depending on the environment that it's in, those methyl groups come, lock on, or leave. But some don't ever leave. Is that a correct statement?
Yes. Yeah, that is correct. A great example of that is in females, where the sex hormone is the X hormone or the X chromosome. One of those X chromosomes will be silenced using DNA methylation. So it plays a big role in regular function. And when that doesn't happen, I can't remember the name of it, but there's a disease that occurs if that does not happen.
Do you know at what stage of the embryo that X chromosome methylation happens?
Gosh, yeah, that's a good one. I don't know. Do you?
It was a chick question. Of course I do. It's in cats, at least, it's in the 16 or 32 cell stage
Oh, nice.
of female chromosomes. And here's the really cool thing. It's how calico cats get their coloration.
Yeah, so depending on which X in the female cat is, it could be either X, you have 2 copies, one from mom, one from dad. And so which X in which cell in which area is being silenced results in which color you see in the calico cat. So that's why you get that patchy coloration.
Yeah, and I don't know how this happens, but I have never seen a calico cat that doesn't have the septum of the nose divided right along the middle.
Really?
It
haven't
doesn't
noticed.
seem, it's supposed to be random. So like at the 16 or 32 cell stage, the X chromosomes randomly get methylated. But for some reason, I'm almost always, I don't believe I've ever seen an exception. I'm sure someone's going to send a picture in that says, nope, my calico cat looks just fine that way. But they almost always seem to have a septum differentiation at the face, which I think is just fascinating.
That is.
Also,
oh my gosh.
So the listeners obviously hear this. Female cats have 2 X chromosomes. Male cats only have 1 because they need an X and a Y to be a male. That's why you'll never see a male calico. There have actually been a couple documented, but they're intersex. They're actually biologically male cats that are genetically female, which is, again, super awesome.
Yeah, there's those rare cases.
Okay, so we've established like what methylation is, sort of talked about what epigenomics is. We've talked, and then some genes get methyl groups placed on and off, depending on what's going on in the environment, because those genes need to get turned on and turned off. But some methyl groups get put on the DNA and they act like a dead switch and they'll never come off. Tell me about methylation and aging in the genome.
Yeah, so one of the interesting things with methylation and aging is the maintenance system in the body. So regular maintenance inside your body, you have cells that need to replicate just to maintain body function, right? And during that process, and you make a new copy of the DNA code, you need to place those DNA methylations back on there after it's made. And so over time, we tend to see a loss of DNA methylation just through mistakes, and it not keeping up and keeping those methyl groups there. So overall, with age, you see organisms losing or this hypomethylation that occurs. And this is random, like mistakes that are happening over time. And that's really interesting. But at the same time, DNA methylation could be more precise. When you're young and you're growing and dividing, you might have certain genes that need to be on and expressed more for that cell division and that growth in the organism. And then later in life, you don't need them, so you might shut them down. So there's different ways and mechanisms and things that we can pick up on when, say, we want to use DNA methylation to estimate age. We can tap into those mechanisms, both the random and the more precise.
So let's talk more about that, because obviously this is where we're going. This is what the product that EpiPause sells now. And it's also probably the product that people who are interested in longevity for their pets are most tuned in on. In my mind, there's always been sort of two scenarios why I would want to use an epigenomic age test. The first and probably most common one is I just adopted an animal. I don't know how old it is. The people at the shelter said it's blah, but honestly, as a veterinarian, we know that's a guess. There's no way we can estimate. In horses, you have some estimation based on dentition, but even in dogs and cats, if they're over 6 months old, there's really no way to definitively say based on anything that you can see. So that's probably why people usually want to use age clock. I think the second is also interesting, which is, well, maybe there's a third, and that's the people who are testing to see if they know the age of their pet, and they're just testing you to see if you're right.
Yep, that's another one.
But maybe the second one, just to give everybody the benefit of the doubt, is my dog. I know my dog is 8 years old. I wonder what its DNA tells me its age is. And if that's dramatically different than its chronological age, that could be kind of interesting, either ahead or behind. I want to get to chronological versus biological aging later, but let's just expand on how does a chronological age test work? I just adopted a dog. I don't know how old it is. I send its cheek swab sample to you, and you come back and tell me based on what its the age is.
Yeah, so we have a couple of markers in the genome that we found to be really accurate to chronological age, and just chronological age. It doesn't seem to be affected too much by health issues that they might have or any other environmental variables. And so we've kind of tapered down how many markers we want to use, so we take out that environmental noise. And we use those exact genes to estimate age, and we just measure, and it's a linear equation. It's either gaining DNA methylation or it could be losing. And we have a few markers that we use to estimate the age for an animal.
When you say a few, are we talking like dozens or scores? I know it's probably proprietary and I wouldn't ever ask the exact number, but are there bunches, or have we really figured out that if we just based on a few, we do know?
Yeah, for us, we found out that we could use under 10, I'll say it like that, under 10 markers and be very accurate. And there's a lot of literature that has done chronological age clocks, and they've done like hundreds, they use hundreds of genes. But what we found is that there's a lot of environmental noise when you're using that many markers. So we prefer to use less.
That's really cool. And how did you validate that?
Yeah, so this was how I started the company. I started going to dog festivals locally and asking people if I could swab their dog and how old their dog was. And so I generated this big data set of known age animals where they knew their birthdays. And we created the model based on them. And we had dogs from little Chihuahuas to big Great Danes. And we found out that we were able to make a model that fit and was very accurate across all different size dogs. There is, of course, some error around that estimate. But we typically see within 12 months of actual age is our accuracy.
That's great. I mean, and also it conjures up this really funny idea about somebody at dog parks in South Florida asking everybody if they could swab the dog and how old they are.
And I still do that from time to time.
About how many dogs did you have to use for the validation study?
We had a few hundred. And actually, we started out with like a really small group. Like our first sequencing event we did was like 80 animals. And honestly, the accuracy did not improve too much from adding in more samples. It was really robust off of a small, which is interesting but expected from the work I did in my PhD. I created an age estimation test for dolphins during my PhD, And that model was built on 40 dolphins.
Well, first of all, where did you find 40 dolphins?
Good question. So there's this really cool population of resident dolphins in the Sarasota area in Florida, Sarasota, Florida. And there's a group, a research group over there that has been watching them for over 50 years. And a dolphin's average lifespan is about 50 or so years. So they had animals that they knew from birth at all different ages, so we could have our known age population to work with.
Oh, how cool is that? And each animal was identifiable through phenotypes, right? So like you and I might, well, I would look at a bunch of dolphin and go, wow, that's a bunch of gray dolphins. And you would look at them and say, there's a bunch of individuals.
No. I look at them and I'm like, who knows? Those researchers know them well. I mean, they have the photos. They go off of the fin. It was interesting. They have pictures of all the fins, and there is little notches and things that they just know like is unique. It's like a fingerprint. It's cool.
Yeah. I went, quick aside, we went on a great white shark diving expedition years ago off Isla Ensenada, which is apparently no longer allowed there by the Mexican government, but it's 200 miles off the coast off the west coast of Mexico. And what was shocking to me was that as soon as you got on board, you got a notebook that had 200 different sharks identified just by scarring patterns or fin notches and things like that. Again, clearly you understand people who do this all the time really do know their animals. So you had a population of 40 dolphin. You had researchers who could identify them and tell you exactly how old they were. And the obvious question, which everyone's going to ask, is where is a dolphin's cheek?
I love it. So with the dolphins, we had to use a different sample type. So with dolphins, they're really hard to capture. And I've been on an expedition where they like catch them and work them up and get all the different samples. It takes like hundreds of people to do this, to stop a dolphin and take it out of the water. So the most common way they sample dolphins is through biopsy darts. They'll shoot them, and it's just a little tiny, it's like 2 millimeters, I think, or 2 to 4 millimeter biopsy darts is what they use, something like that in that range. It just sticks in and falls out with a little chunk of muscle. So that's what we used.
Gotcha. Very cool. Very cool. And so you created an age clock, chronological age clock for the dolphin during your PhD work. That was a Florida International. And then you decided at that point, boy, it'd be a lot easier to get cheek swabs from dogs than biopsy darts. And my bet is the commercial reality is probably that people are willing to spend a little bit more on their dogs than their dolphin, and there are probably a few more. At least the numbers work out that way.
That's pretty much what happened. I actually, I didn't know what I wanted to do when I was finishing my PhD. So I went to this commercializing research workshop to see, some options of what I could do outside of academia. And while I was there, I was telling them about, I can age dolphins. I can age any animal. I can make a test for it. I'm going to start a lab doing that for all these researchers because no one knows how old the animals are in wildlife. And then I was like, and I was talking to business people, right? And I'm like, oh, I can also do it for dogs and cats. And their eyes lit up and they're like, look into that. And I was like, all right, there's this huge market for dogs and cats and no one's doing this. And they're like, just focus on pet for a little bit. So that's what I did.
That's awesome. All right, just a quick one before we carry on. If you're enjoying this conversation, hit follow or subscribe wherever you're listening. It really helps the show grow and lets us keep bringing on great guests. Okay, let's get back to the conversation. I want to focus a little bit on the biology. So you've identified, let me just kind of recap where I think we are. You've identified a kind of handful, less than a dozen genetic markers that are either methylated or not methylated?
Yep, increasing or decreasing.
How can you do that with a single sample? Maybe I'm just missing.
Oh, we do sequencing. I think that's
so you sequence it.
Yeah, sequencing.
You sequence the sample, and the methyl group that you're You're looking at the number of methyl groups that are in those 10 areas or less than 10 areas?
Yeah, so the type of sequencing we do is a little bit more evolved than just genetic sequencing. So with genetics, which people might be more familiar with this and have done some of the genetic sequencing that's out there. Sometimes it's a dry swab, sometimes it's a swab put into liquid. Um, all you're looking for with genetics is a snippet of a sequence to identify what kind of gene type they have for each gene, because you have different types or alleles, we call them, for different genes. And depending what allele you have, you might have a predisposition for disease or something like that. With what we're doing, we're less worried about what that DNA sequence actually is, but we want to know how much DNA methylation is there. So that's our main focus, and we treat the DNA with some chemicals that allows us to visualize those cytosines that have methylation on them. So we do this fancy treatment, and then we sequence, and then we can see, okay, there's methylation at all these places in this particular gene, and we can add it up. And really what we want is when you take that sample, you have several cells, not just one. You're taking several cells, and we calculate the percent. So out of all those cells at this particular gene, how many of them had how much DNA methylation? We turn that into a percentage at a certain gene site. And that's what we use in our model, is the percentage that's there.
Gotcha. So the percentage of methylation will tell you, then you can put it on this linear scale, your percent, that means this age. That's really cool.
Yeah.
So let's segue on to biological age. So there are lots of tests out there in the longevity world around your biological age. In the human side, it's usually a kind of a long algorithm of different biomarkers and how that tests against different parts. My watch tells me that I'm usually, congratulations, you're 17 months younger than you really than your age is. So tell me how that works in the methylation world.
So when someone says biological age, they're referring to this idea that you might look younger or older than you actually are. And that could be the same for your dog. And it's just like a health metric that we understand instantly, because when you say, oh, this person looks like they're 80 versus this person looks like they're 40, that means something to you. You understand it. So that's why we would calculate a biological age. And now there are a million different ways you could calculate biological age. So that's the fun part. So when your watch is telling you biological age, it's going off of other markers and there's a reference range for those markers that they equate to being as, this is what the average 40-year-old looks like with those markers, and the average 50-year-old looks like with those markers. And maybe they have some standard of a healthy 40, a healthy 50, right? So with DNA methylation, it's similar. That's what we're working towards.
How does DNA methylation work with biological aging?
So with DNA methylation, there's a couple different ways we could calculate biological age. So similar to chronological age, there's several markers across the genome that we see changing over time with chronological age. And we can look at that and figure out a reference range, like a normal, like this is a healthy 40-year-old, a healthy 50-year-old. This is what their markers look like at this set of genes as they age. And then there could be certain individuals who are aging or aren't as healthy. So an unhealthy 40-year-old has a different pattern at these markers. They've accelerated and went faster. They look more like the 50-year-olds. That's one type of model that could be created with DNA methylation. Another idea around biological age is that there should be certain patterns of DNA methylation. And when you're heading into a disease state, it falls apart. It doesn't look normal. It's abnormal. It could be chaotic. It's not necessarily that aging pattern we expect where it's increasing steadily or decreasing steadily. So there's different patterns that we could use in a biological age model.
Are there examples of diseases where that's really understood?
So a good one would be cancer. So especially if you're looking at cancer in the tissue where it's happening, we see a very distinct different pattern that's correlating to that cancer disease. So that one's a really robust one. The problem with getting to that is that it is tissue specific.
It's those cancer cells that are displaying different methylation patterns that,
Yes,
okay, yeah, interesting.
exactly. But then they are able to detect a lot of cancers in the bloodstream. They're
Right.
sloughing off of those cells into the bloodstream, and they can be picked up in cell-free DNA. So there's a couple of really cool, like Grail is doing a DNA methylation. It's DNA methylation plus proteins that they actually, it's a combined model to detect cancer in the bloodstream.
Detect cancer anywhere, but it's from DNA that's floating cell free in the bloodstream. Very cool stuff.
Yeah, it's very cool.
So, unless you're talking about a tumor in the cheek, my guess is that most of the time you're not picking up those kinds of patterns in a cheek swab sample. So, you're looking more at an overall variance in variations in methylation when you start thinking about biological age tests using methylation.
Yeah, so for us, that was the big question. Is there's all these we see in human, we look out at what the field has done already for biological age with DNA methylation, right? And we see all these different models for humans focused on different end states. Some are focused on disease development, others are focused more around that chronological age model and just those typical markers that change over time. Um, and so for EpiPause as a company, when we start thinking about biological age, we want to make sure that was something that would be useful in general to pet health and where pet health stands today, which is really being reactive. We'll eventually get to a point where we're more set up and we can do more proactive things. But our first step should maybe be around what we're familiar with and we can act on, which would be disease states. So for us, we actually took an approach where we looked for markers that were really had a change when a disease is present, and decided that that would be our first biological age model to tackle. Um, so that's actually where we're starting as far as that spectrum goes of all the different ways you can calculate biological age.
So, any disease state? Are these sort of generalized disease states, or are they specific diseases that you're looking for?
Yeah, so we went general first. We wanted to see if we could just have a well-rounded catch anything kind of. We named it unhealthy, the category, so it's healthy versus unhealthy, and we threw everything in there. We have cardiac disease, we have liver functions, we had CKD,
Chronic
allergies,
kidney disease.
cancer. Yep, yep. Chronic kidney disease. We even threw allergies in there.
Wow.
And we made that our unhealthy group. And we were able to make this prediction model of how likely are they to be unhealthy and have one of those diseases. And we were able to create a model with a handful of markers. So we have like, let's say it's less than 500 gene markers in this model.
Wow. Very cool. And so, the logical reason you would want to know your biological age or your pet's biological age is so that you can do something about that. And then if they're biologically older than they should be based on when they were born, you can make them better. And so I assume that your disease state, if you correct that disease state, potentially you would get closer. Is that a correct assumption?
Yeah. So I like to talk about the big vision for it. So what we're aiming for with this biological age is learning how all the different products and diets and things affect that aging trajectory and learning more about how that aging trajectory leads into disease. The more we know about that, the more we can course correct before we're ever developing a disease So that way we're working to avoid disease development in the first place. It's kind of audacious, big goal, but that's ultimately what we want. And if you can solve for not developing the disease in the first place, that's where you really move the needle on longevity, because you're preserving that health span. So that's our major goal. And so where we're at today, especially for pets, is really early. We're in these early stages that we just know if we don't measure, we'll never get there. And so we created, a nice big panel that captures a lot of genes so we can monitor and watch this DNA methylation and figure out which genes really matter the most to be watched. And eventually we'll get to a point where we have the ideal model to catch diseases early on, the ideal model for when we want to change diet and optimize diet for a pet. There's all these different use cases for this kind of information.
And this is non-tissue specific. So you're saying I can take a cheek swab from these dogs, and eventually the vision is that a cheek swab, the methylation pattern that we pick up on these several hundred markers, will help us understand if this dog is headed towards not just any disease, but specific diseases.
Yes, exactly. That is our goal, is to, now that we have a way that we can identify or predict healthy versus unhealthy, as we grow the database, our main focus is to try and categorize by organ group, because that gives us something more specific. So if we can identify that these are genes related to health of kidney, which we can see in the mouth, there's a systemic effect, and that's why we can detect these things in the mouth. If we can identify that, or the heart, or whatever, that gives us something actionable, where you go into a vet and you tell them that you want to help support your dog's heart. Well, okay, there's supplements, there's things, well, what would you do to help support the heart if it had a disease? We can start those things earlier and see if we can avoid that disease actually developing. We have to learn how to avoid the disease, is the other part, once we detect what organ group. So there's a whole lot of learning to do.
It's the ongoing chicken and egg question about, do diagnostics come first or do treatments come first? And the short answer is diagnostics usually have to come first so that we actually know what it is that we need to treat. But there is a frustrating point here where you will find out we've got kidney disease in an animal, but we have nothing that we can possibly do about it. That'll be that's a phase, and that's challenging. Oh, so this is fascinating. So I think eventually we will get to this holy grail of these are the things which keep us healthy and out of disease. There's always, of course, going to be the randomness of infectious disease or trauma. But these are systemic diseases which are somewhat spontaneous, potentially avoidable or not. And we'll be able to start seeing early indications of them just in the methylation patterns inside the cells. Absolutely fascinating. Andrea, can you give us a timeline on when you think this stuff is going to happen? I mean, obviously yesterday is the answer that you'd like to see, but what's more realistic? When do you think these kinds of tests will start coming onto the market?
Yeah, so for us, we've already launched a biological age for dogs. We plan to launch one for cats this summer. And this is the V1. This is the MVP stage of it. Like I said, if you don't start measuring, you'll never get to that end stage. So we're really looking for these pet owners who might come along on the journey with us and know that they're building a baseline. So here's where baselines, I'm going to go on my soapbox about baselines for a minute here. So just like you take your vet, or you're supposed to take your pet to the vet every year and do blood work, they're building a baseline so they know what's normal for your pet. We're all individuals. Our dogs and our cats, they're individuals. They have different baselines for different things. Same thing with DNA methylation. So if you start measuring across their life and you learn what's normal variation for them, that's where you become sensitive to picking up a health issue, and really early. So it'll be trickling along. The DNA methylation looks normal here, and then all of a sudden it starts getting higher and higher and higher at this one gene. You look at what that gene is functioning in, and it has something to do with kidney health. maybe the kidneys need some more support, and that's where you're catching the disease early on, or maybe it's no disease and you're just catching something and avoiding the disease. So that's why we decided it was really important to go ahead and put this out to the world. And it's really more so than what we can tell you now about DNA methylation, what that means for their health. There's only so much we can tell you at this moment, but that becomes a record. We've captured that DNA methylation time point for your pet, and we can build on it, because then you'll measure again and we'll see how much did it change. What things have you noticed? Have you changed their food? Have you changed anything? Have you not? And then you have this nice baseline. So it's really important that people start measuring right away. And this was my whole scientist creed. I used to go around to science conferences when I was doing wildlife and be like, get the sample, whatever it is. You don't think you need a sample, get it and preserve it, because then new technology comes out and you want to go back and see. And unless you got that sample, you can't. So that's my whole soapbox is you got to get the samples. And that's my big push to everyone. Start sampling, getting the samples.
The obvious analogy is weight, right? I mean, you don't know if you're gaining or your pet is losing or gaining weight if you don't take the pet's weight.
Right. Yeah.
Relatively, on a regular ongoing basis. I know my dog weighs 47 pounds, and if I go in and she weighs 52 pounds, well, that's kind of significant. That's a 10% weight gain. So I see the logic. Great. I think of course, logistics and cost are always going to be factors for pet parents. Like, what are we talking about for these kinds of tests? At least at EpiPlus.
Yeah, no, that's true. And that's exactly where my head was going with like my soapbox and telling people to sample. Yeah, there is a cost to getting it sequenced. So I might need to start like a biobank system that's cheap and you just like stock up the samples or something, but maybe we'll think about that one. But yeah, to actually get a sample sequence, it's pretty expensive right now. I'm trying to figure out how do we lower the cost of sequencing so that it's more available to more people. I mean, what use is it if no one can afford to do it? And that was my whole shtick at wildlife, because we're talking about conservation and people are boots on the ground saving animals. How are they going to allocate money for these kind of tests and these kind of tools? And we have to make them affordable. So I am pioneering that and working to lower the cost to the end consumer. But at the moment, like our test, we put it out there, it has a retail price of $250. We're measuring thousands of genes, though. I mean, you're making a really nice baseline for your pet, if I can make that argument and convince you.
And so retailing at $250, what you're not talking about thousands of dollars every time, and we're not talking about whole genome sequencing or anything like that. And you do establish a baseline, and you also can get some value out of that. I mean, this is really exciting stuff. Someday one can imagine having every 6 months testing the biological age of my dog, monitoring it like I do with every other health metric, and maybe even getting early indications of diseases before they ever become clinical, which of course is the goal. Andria, this has absolutely been fascinating and super fun. Is there anything else that you want to talk about? I do have my always question, but anything else that we didn't chat about that you'd like to hit before we leave?
About those samples and getting lots of samples and how important that is to us being able to actually give some valuable insights and not beyond the baseline. The baseline is really important, but beyond the baseline, people want to know more insights. So we have this huge goal to collect 10,000 samples in the next 18 months, which is a lot of samples. And so we invite people to also join in and submit an R&D sample, which can help us get that goal. So definitely visit us, see about joining us in research efforts, which is free to participate in.
Awesome. Well, thank you for that. So last question I ask all the guests. If this topic is really resonating with some of the listeners and they just want to dive in deeper and they don't need to call you directly, what are 2 references that you might point people to just to get better background on this area?
So 2 references. Well, one, We are trying to put information on our website that might be useful for people to learn more about longevity in Healthspan and these types of DNA methylation related tests and things that are coming out for both humans and people on our website. So we're trying to be a good resource for that. But other than that, there is a book out there. You probably know it. I'm drawing a blank on the name, but it's Peter Attia's
Outlive.
Outlive. There we go. I was going to say, I kept thinking Alive was popping up in my head. I'm like, it's
really
not
close.
that. Outlive. I think that book, especially in the opening chapters where he talks about diseases taking root and starting way ahead of time, really resonates with our goal of why we want to monitor and watch things and identify them before they're actually a disease state. It pairs nicely. It gives a nice visual, and he gives really good examples as a physician for humans. So I think that's a really great book to help people kind of wrap their heads around this.
Yeah, he articulates some basic concepts in that book really exceptionally well. Well, awesome. Thank you. We'll link both of those in the show notes as well. Andria, it's always a joy to talk with you, and really appreciate you sharing all this knowledge and coming on the show today.
Yeah, thank you for having me. This was fun.